Mike Thiel is a second-year master's student in Integrated Biosciences program at the University of Minnesota-Duluth. His research interests lie at the intersections of avian ecology and toxicology, with the ultimate goal of protecting wild bird populations from environmental contaminants. Mike.
Sweet. Thank you, Clinton. Thanks, everyone, for being here. I'm excited to be here and talk to you all about some of my thesis research. Before I begin, though, I would like to answer the question you're all probably wondering. And no, I am not closely related to a bald eagle. I am just a bald man. So with that being said, I'm going to dig into some of my master's research here. Here's a brief little look at an outline of what we're going to talk about today. I'll start with my research question as a way to kind of introduce my study. Keep that in the back of your mind when we start going through some background. I'll talk about what PFAS is and some of the things that we know it does. And then I'll also talk about avian ecotoxicology as a field, and as well as some previous avian PFAS research that's kind of led us to where we are right now. Then we'll talk a little bit about my research. Just want to note right now I'm kind of in the middle of my research. I'm a little over halfway done with my master's. So I've got some preliminary research to share, some analysis, and we'll talk about my objectives, my hypotheses, methods I've used to address those, and then wrap up with some conclusions and next steps. So here is my primary research question. Broadly, we want to know, does PFAS exposure have a significant effect on the metabolism and stress response in developing wild birds? Just out of curiosity, could I see by a show of hands, how many of you have heard of PFAS before? Okay, that's great, that's a lot. If you didn't raise your hands, don't worry, I will tell you now. PFAS are per- and polyfluoroalkyl substances, or PFAS. And when we talk about PFAS, we're not actually talking about one or even two chemicals. It's actually a class of thousands of chemicals. And the EPA has actually identified over 7,800 of them in the environment right now. And all of these chemicals have varying levels of toxicity depending on their structure. All of these chemicals, though, do have shared characteristics that give them unique properties. They are all fully synthetic. In fact, PFAS didn't exist 100 years ago. It's completely man-made. And some of those unique properties include a carbon and fluorine bond. Whoops. The carbon and fluorine bond here on the, whoopsies. There we go. The carbon and fluorine bond you can see here on the top right between the gray circles and the green circles. And that's really important to note because that's one of the strongest single bonds in all of chemistry, so it gives it some really unique properties. Also, these tend to be what we call amphiphilic or surfactants because one side of the chemical is hydrophilic while the other side is hydrophobic. And it also means that they're really ubiquitous in water, and they're globally widespread at this point. Essentially, they're transported through the entire water cycle. And interestingly, the CDC found that these chemicals were present in 98% of human blood samples taken from 2000 to 2014. So here's a little history of PFAS, how we got to where we are right now. Back in the 30s, PFAS was actually accidentally discovered and named Teflon. In the 40s, we saw it being used in the Manhattan Project. In the 50s, it started to be used in different industrial settings and in consumer products like nonstick pans. In the 60s, AFFs were produced, or aqueous film-forming foams. This is an important one to note because this is a major source of environmental contamination of PFAS. In the 70s, we started to see the use really expand across a lot of different industries. NASA even started using it. And in the 2000s, we started to see a voluntary phase-out of PFAS production in the US because we started to notice some adverse effects on human health. There were links found to cancer and some thyroid diseases. And present day, we still have new PFAS being developed, but we also have an increased public scrutiny and with that, an increased effort to regulate these chemicals and an increased focus on the environmental impacts. So a lot of times when you hear PFAS, it's also coined forever chemicals. So how did it earn that name? Well, we know that it's extremely persistent in the environment because of those unique properties I mentioned before. It's really chemically and thermally stable, so for a lot of these chemicals, there's no natural route of degradation. Also, we see that it bioaccumulates and biomagnifies within an ecosystem. So the bioaccumulation is essentially just the buildup of a substance in an organism over time, while biomagnification is the extension of bioaccumulation through trophic levels. One interesting thing to note is that PFAS we see adheres to proteins, whereas a lot of other bioaccumulating chemicals we see is what's called lipophilic, so it likes to bond to lipids and fats. Here we can talk a little bit about avian ecotoxicology. This is the field that is primarily responsible for measuring these effects on birds. Ecotoxicology is just simply ecology and toxicology. It's at that intersection. Right now, there are some gaps in avian ecotox research. For example, the vast majority of avian ecotox research is focused on the clade gallo-anseres, which is circled here. This entire circle is all of the diversity of bird species, and gallo-anseres only make up about 5% of all bird species, and around 90 to 95% of toxicology studies are on gallo-anseres. Also want to point out that passerines, like tree swallows, are especially underrepresented in the literature, even though it makes up over half of all living bird species. So this underrepresentation of bird diversity in toxicology studies makes it really hard to extrapolate results to the entire diversity of bird species. And Rachel Carson actually noted this in 1962 when she said, "Chemicals may have been pre-tested against a few individuals, but not against living communities." That was 60 years ago. We still see this problem. We've come a long ways, but it's still definitely an issue. So let's talk about avian PFAS research thus far. So far, we do see that laboratory testing is limited. Right now, there's laboratory-based literature on chickens, bobwhites, quail, cormorants, and herring gulls, and that's only a handful of about 10,000 species. Field research so far has focused on seabirds, which makes sense. They exist at a high trophic level. They're really closely associated with water, and also locally on tree swallows around the Twin Cities and in Wisconsin. And this work has been done in the last decade or so by the Custers et al. So far, all of these results suggest at least one major negative effect within birds, and that is what's called endocrine disruption. So what is endocrine disruption? Generally, we see that endocrine disruptors mimic natural hormones or block receptors from hormones, so it can affect pathways and cause an overactive or underactive thyroid. It's important to note, too, that this isn't always acutely lethal, so we can see effects on the fitness of the bird, but not necessarily the bird is killed. So that makes it a little harder to study. Some other examples of endocrine disruptors that you may have heard of include DDT, organochlorines, PCBs, and dioxins. So what do we know about PFAS specifically as an endocrine disruptor? We see that there is evidence that it influences thyroid function. Many different PFAS chemicals can mimic hormones, including sex hormones and T3 and T4 hormones produced by the thyroid, which ultimately disrupts the HPA and HPT axis. The HPA axis is the hypothalamic pituitary adrenal axis. This is primarily responsible for mediating a stress response. The HPT axis is primarily responsible for regulating metabolism through the release of T3 and T4. Here's a quick little diagram of how the HPT axis works and how it might be affected by PFAS. So we see the hypothalamus in the brain releases TRF when it's time to stimulate metabolism. TRF is thyrotropin releasing factor. That interacts with the pituitary gland, which then creates a thyroid stimulating hormone, which, as the name suggests, stimulates the thyroid to start making T3 and T4. Now, normally T3 and T4 will enter the bloodstream, where it goes into the liver and into bile and regulates metabolism. And then when there's an abundance of T3 and T4, a negative feedback loop forms, and it essentially shuts down this situation so that metabolism doesn't spiral out of control. So one possibility for PFAS disrupting this axis is by some PFAS mimicking T3 and T4, the hypothalamus could potentially detect this and think that it needs to shut down the system again. There's also other pathways where PFAS might affect transport proteins or any other proteins in this, depending on the chemical and depending on the species. But essentially, this results in a much lowered metabolism and also a lowered stress response. I'll talk a little bit about my research. The general purpose of my study, as the research question suggests, is to explore the adverse effects of PFAS on developing tree swallows, eggs, and nestlings. We're going to do this by looking at metabolic rates of eggs and nestlings, as well as stress hormone concentrations. Overall, we want to provide results that can hopefully inform regulatory decision-making. Hopefully, we can use these results to compare against lab toxicology results and compare those against those extrapolations, see how accurate they are. Also, we want to observe some of those sublethal effects and see if fitness is still being affected even if it isn't outright killing the birds. We also want to utilize some new methods in avian ecotox, what we're calling field respirometry. And also, last but not least, we would like to have fun while doing it. Here's a look at our study system. This is actually in collaboration with a much larger EPA study up in Duluth at their lab. We have 6 different study sites around the Duluth area. Each of these sites has varying concentrations of environmental PFAS contamination. Most of this contamination has come from the airbase site here in the middle. For years, AFFFs were used in firefighting training exercises and has gotten into water sources. Each of these sites also has 20 to 40 nest boxes that we've put up between 1 and 3 years ago to try to establish a good colony. Here's a look at what some of our sites look like aerially. This is Rice Lake on the top here. All of these blue pins are individual nest boxes. As you can see, we try to get it really close to water. Tree swallows really like nesting by water. And we try to spread it out a little bit. And here's a look at what the actual nest boxes look like. They're just a pretty basic bluebird design nest cavity box. So why did we choose tree swallows as a model species? There's a few really good reasons. For one, they do readily nest in artificial nest boxes. So we can place these nest boxes up at sites that we know have contamination, and generally they will use these boxes. Also, they're what are considered kind of loose colonial nesters. If you set up some boxes at a site, generally the first year you can expect to get maybe a few nests, and incrementally that will increase every year after that. Also, we know that these birds feed within a really localized area during the breeding season. And they primarily feed on emerging aquatic macroinvertebrates. That's important because we know these invertebrates are really heavily exposed to PFAS in the sediment and in water. Also, tree swallows are relatively easy to work with. They're not quite as bothered by researchers as maybe black-cap chickadees would be. And why do we take measurements on eggs and nestlings? Well, we know, for one, PFAS likely disrupts development of eggs and nestlings. So if we take measurements at key developmental stages, it can give us important clues as to what pathways is being affected by PFAS. Also, we get really reliable exposure data. We know that these birds are being parentally fed locally of those invertebrates that are exposed to PFAS. If we were to sample and take measurements from adults, it would introduce a sampling bias because a lot of those adults might not make it to adulthood if PFAS is having a lethal effect, so we wouldn't be able to really pick up on that. And here's a look at my two hypotheses. My first hypothesis is that PFAS exposure negatively influences tree swallow development, and that effect is measurable in lowered metabolic rates of eggs and nestlings. Hypothesis 2 is that PFAS exposure is negatively related to stress hormone concentrations in tree swallow nestlings. And these are the methods I'm going to use to address these hypotheses. For one, we're going to be nest monitoring at least twice per week, sometimes 3 or 4 times a week, depending on the time of season. This helps us accurately determine the nest initiation dates, incubation onset, and predict those hatching dates. Then, when those clutches are complete, at least 1 or 2 eggs from each clutch are collected for field respirometry at day 10 of incubation, and then they are analyzed for PFAS concentrations. And when the eggs hatch, 1 nestling per brood is collected for field respirometry at day 12 post-hatch, and then analyzed for PFAS and stress hormone concentrations. Here's a look at our respirometry methods. First, we set up our tailgate lab, and then we start working with our Qubit Q-Box field respirometer. Whoops. So the way this piece of equipment works is essentially it takes in outside air, puts it in through the gas pump and a flow rate monitor. From there, it goes into this animal chamber on the bottom, where we either have eggs or nestlings. Then the air is pumped out of that, goes to a CO2 and O2 analyzer, and we're able to get CO2 and O2 concentrations in real time as the organism is respiring. Here's a look at what the raw data looks like when we're taking measurements. This is a run that we did on eggs, and this utilizes the technique called stop flow respirometry. On the y-axis here, you can see CO2 in parts per million, and on the bottom is time in minutes. The way this works is air is put through the chamber at a constant rate, and then flow is cut off from the chamber for a set amount of time. We do it for 3 minutes. And then after those 3 minutes are up, we flush the chamber with the outside air again, and you see this big spike in CO2. And this is essentially the amount of CO2 that was produced during those 3 minutes. And we use that to estimate what's called VCO2, which is the volume of CO2 produced over time. Nestling respirometry is a little easier, a little simpler. This is using open flow respirometry. We just have a constant rate of air going through the chamber. We open up the chamber, place the nestling inside for a set amount of time, and then we take it out, and we're able to estimate VCO2 from sort of an average of the CO2 values over that time. Now I'll talk a little bit about my preliminary results, some initial results from last summer. Here you see this is all egg respirometry from last summer. On the bottom here, I have all of our sites, and this is ordered from most contaminated with PFAS to least contaminated. On the left-hand side is VCO2, and it does appear that there may be some sort of association between lowered VCO2 and environmental contamination of PFAS. Again, this is all very preliminary, and we have a sample size of 38 right now. Here's a look at the nestling respirometry. Not quite as strong of an apparent relationship, but we'll have to dig into that further. So just wrapping up here with some conclusions and next steps. We do see that there's an apparent relationship between VCO2 and eggs and nestling respirometry. A linear regression analysis shows that time of day, mass, temperature, and season do not influence VCO2 for the eggs, but we do see that temperature does affect VCO2 for nestlings. Next steps, we're just waiting for those individual egg and nestling PFAS concentrations. That's when we'll really be able to dig into the data and dig into some analysis. And then there's going to be another field season this summer in 2023 where we'll hopefully at least double our data set. And ultimately, it would be really cool if we could see these techniques used for different contaminants and stressors as well. It doesn't even have to be contaminants, but just any time a metabolism may be affected. And with that, I just want to say thanks. Thanks to my advisor, Matt Ederson, who's going to talk about Hawk Ridge later. Also to the field crew and the Integrated Biosciences program at UMD.
Thanks so much, Mike. Any questions out there? And again, just a reminder, name before your question, and we'll get through everybody here.
I'm Roger Everhart, and I was wondering about the impact of PFAS on the actual macroinvertebrates that are coming out of the water. Is there maybe a side effect of less food that could affect metabolism?
Yeah, that's certainly something that would be really interesting to look at. As part of the larger study, we are doing sampling on invertebrates as well and getting kind of some background diet concentrations of PFAS. And I know the EPA is also looking into effects on invertebrates specifically. Most of those are looking at lethal effects, but yep, that's definitely potentially a cause of an effect.
Hi, Dwight Smith. When you talk about endocrine disruptors, one of the first things that comes to my mind is reproduction rates. Has anybody done research, or are you aware of research having been done on reproductive rates as a PFAS or other environmental chemicals?
Yeah, absolutely. Some of that preliminary research that I talked about, especially by the custers with tree swallows, did find an association in one of the studies between egg hatching success and PFAS concentrations, I believe. So that has been looked at, and that's helped to kind of spur this research.
Mike Menzel. It's my understanding that the Minnesota legislature next year will be looking at legislation to ban the sale of any products that contain PFAS. It will be a small start, but hopefully will yield some positive results.
Yes. Next year, I believe it'll be up to states will start to set their own regulations for PFAS production. And I know that in Europe, a lot of PFAS chemicals are already outright banned.
Any last questions floating around out there? All right. Thank you, Mike.
Thank you.